we may have some options here by something that nature has invented, and that's a biological method of control. There is a method of control that is biological, but it needs work, and that's where our research project comes in. What is this biological option? Dan Kegel, after he had that big problem in 2007, really decided he didn't want to grow wheat anymore. But if he had continued to grow wheat on his land, after a span of 3 to 7 years, he would have seen a very interesting phenomenon. The disease would come under control all on its own. Of course, he would have to keep losing that $65,000 every year, but he would see that the disease would start to come down, and his yield would start to go up, and therefore he could start making money from his field again. This process is called take-all decline, and take-all decline happens just from growing wheat in the same soil year after year. It's a biological process, and it's a spontaneous decrease in the incidence and severity of take-all. How does this happen? That's the research we're doing. So a key thing to notice in this slide is that the soil is said to be conducive when there is disease and suppressive when the disease is under control. So conducive means conducive to the fungus, not to us, and suppressive is suppressive to the fungus and good for us. This is seen whenever there is monoculture of wheat or barley for many, many years. Okay? So how does this function? So farmers have known about suppressive soils for as long as they have been doing agriculture. They did not know what the basis of that suppressiveness was, and that's where agricultural scientists have been analyzing the underlying mechanisms of suppression. We can divide suppression into 2 different types. You have general suppression. This is sort of what happens in your yard when you add fertilizer to your yard. You increase the total number of living organisms that are in the soil. And that's what microbial biomass means, is that the total number of living organisms that are in the soil. And you can do this, as I said, by adding fertilizer, that is organic matter. And how this functions is that basically there are so many things competing with the fungus to grow that the fungus is sort of outcompeted. Okay? And you can see that effect. You know it's biological because you can destroy that effect by heating it to 70 degrees. This process, this general suppression, is not transferable. So that becomes relevant when you look at the next slide, which is where we talk about specific suppression. So specific suppression is superimposed on top of general suppression. This is a very different phenomenon. This is a very different process. So how does this work? If you remember, in general suppression, it was a competition between all of the living organisms in the soil and the fungus. Here, one or a small group of microorganisms actually interact directly with the fungus and prevent its growth. And there can be different mechanisms by which this happens. How do we know the difference between general and specific suppression? One key factor is that specific suppression is transferable. What does that mean? We'll see just in this slide and in the next slide. So if you look at this slide here, you can see that on the far left there is the check or the control plants. And how these plants were generated is you actually grow the plant in the presence of the fungus. And you can see that you have pretty severe disease, the roots are blackened. Now we take soil from conducive soil, so the Quincy virgin soil is conducive soil. Remember, conducive means conducive to the disease, so that's a lot of disease in that time. When you transfer 0.1% of the conducive soil into this environment, the disease actually gets worse, which makes sense because the conducive soil has more fungus. But now if you take the take-all decline soil, that is the suppressive soil, and you add that to the system, notice in the far right where it says the Quincy TAD soil, you can see that the roots are much healthier, right? And the disease is under control. The fact that we can transfer this with 0.1% of the soil says that it's something living because it's now increasing in number and protecting the field. We also know that it's a living system that's allowing this control because you can kill it by heating it to 60 degrees for 30 minutes. So we have something biological that is controlling this disease that we can actually transfer. When you're faced with this kind of question, scientists go to work to understand what is the organism, what is the living organism that is causing this. And if you think about it, this is a huge problem. This is a very difficult question to answer because everything that is possible, everything living lives in soil. Suppressive soils owe their activity to a combination of general and specific suppression. So you sort of stack one on top of the other, and that makes that soil even more suppressive. Two good examples of suppressive soils are shown on this slide, and as you can see from the dates, they are from over 100 years ago, that these two areas of land have been growing either flax or wheat, and you see that suppressiveness continue for that long. In fact, these two areas are under protection and they have to continue to grow flax or wheat on this for the foreseeable future. Scientists like to collect samples from these areas to study this suppression. And the field on the right has been under wheat and is under take-all decline. Scientists have been studying these soils to understand the mechanism of specific suppression. And this research started before all of the technology that we have today with DNA extraction and all of this was available. And this slide and the next slide that I'm about to show you represents work done by many, many scientists over decades to understand how does this specific suppression actually work. So how do you do this? This is a giant problem to solve. This is a really tough question to ask because every living thing that lives anywhere else lives in soil. You have fungi, you have bacteria, you have viruses, you have worms, you have insects. Everything lives in soil. How do you narrow it down? You have to ask very specific questions. You say, if I heat it, it gets killed. So it's a living organism and not something chemical. If we treat it with a fungicide, does the suppression go away? Well, in this case, if you treat it with a fungicide, the suppression will not go away. And therefore, we know that it's not a fungus that's causing the suppression. If I treat it with an antibiotic, does it go away? Well, in this case, if you treat it with certain kinds of antibiotics, it'll probably go away, which tells us that it's a bacterium that's causing the control. So it's a trial and error process of asking questions and working in the lab to answer this. And as you can see, this will take a long time and a lot of effort to elucidate the mechanism by which suppression works. So suppressive soils, the thing to remember is that the pathogen does not go away. The pathogen still stays in the soil, but the disease is brought under control by the organism that is suppressing the disease. When you understand the concept of suppression, you can then apply the concept of biological control. Biological control is the use of one living organism to control another living organism. And this can be done by bringing in new strains or by using the strains that are indigenous or already existing in the soil. Ideally, you want to use the strains that are indigenous or already existing in the soil, because when you bring in new strains, sometimes they can become invasive species or you could have other problems. And it's important to understand how this process of control works. You could have what is called antibiosis, which is one organism produces a chemical that is harmful to the other. It could be because of competition. It could be parasitism or predation, where one literally eats the other. Or it could be done by creating a condition in the plant that makes it resistant to the disease.